it's always a pleasure to be invited to give a talk regarding what we are doing in Singapore and how we can advance clinical healthcare. So when I was really being invited, I really thought about what I can talk to the audience today. I know it's on university, I know it's about medical hub. So I'm actually quite pleased to be arranged right behind Prof. Amal as well as Prof. Chin. So towards the end of this sharing session, so for the lesson learned from the biomarker trials, could then convince both professors that what they're doing in Malaysia is really impactful. It is important to bring in more drugs for clinical trials. It's also important to actually take samples on patients, make sure it's properly being used to advance translational research in Malaysia. So first, I know of interest to be pretty clear. And my laboratory was set up in Nanotechnology University for the past 3 weeks. We are a very new lab. And the focus of the lab is actually very simple. How can we stop cancer from progressing, right? And every time I present this idea to my oncologist friend, they always say, please don't drive us our business. We need a job, right? But essentially, it's like, can we really stop cancer from progressing? We can't, right? And by definition, we claim that cancer progression is a form of evolution. It's really how a normal tissue becomes malignant, how they evolve from stage 1 all the way to late stages, how they react upon treatment, and how they eventually become relapsed and metastasis, right? So these are, every single stages is a form of progression. So at the earlier stage of my career, we focused a lot on how we can tackle the late stage cancer when they relapse, when they go through resistance. And during my late postdoc period, we then try to understand how can we intervene cancer at the earlier stage, how can we identify individuals with a higher risk of cancers, and how can we stop those? And so the key focus of the lab is actually very simple, right? Can we improve clinical prognostication through intrinsic and extrinsic determinants of markers, right? And why do we say so? We know for sure that drug-induced intracellular signaling, how the cells react, how the tumor cells react to individual drugs, basically shape the cell fate. We now know that the ecosystem, the microenvironment, could shape how these tumors react to treatment as well as respond as well. So adding one and one together, we are trying to understand what is the complex microenvironment from the intrinsic cell autonomous mechanisms all the way to the interactions between the complex microenvironment, how these two components basically determine how these individuals respond to a specific treatment. And so, of course, there are 1,000 different challenges that we face in the clinic right now, I mean, as many of the speakers have actually conveyed over the past 2 days. But what we are really trying to do is that can we identify important clinical challenges that we are hoping we can tackle from the lab. So for today's, there will be 2 different case studies that I would like to share with you. So first of it, right, there's really a lack of Asian-centric genome database to advance precision oncology, right, in Singapore, as well as Malaysia, right? So, Prof. Rahman, you are very right about the EGFR inhibitors, right? So we talk about precision medicines, right? We haven't talked about TCGA, the first largest cancer cell atlas that's been trying to profile the somatic mutations, right? So this is a very beautiful review by my colleagues, Daniel. So using lung cancer, it really revolutionized the entire treatment of lung cancer since the day that targeted therapy has been introduced to the market. So you can focus over here, right? There are indeed a lot of driver oncologies that are found in lower lung edible carcinomas. We have NTRK, we have RED, we have BRAF, many others. But one of the very specific cases that we see is the EGFR, proportional EGFR mutations, right? It is significantly elevated among the Asian proportions, as you can see, compared to the Caucasian population, 16%, we have 46%, right? So this really sped up the entire clinical trial development, like what Prof. Arman said. Initially, when they first bring in the tyrosine kinase inhibitors, is it going to work, right? But the case of how somatic variants are actually being found to be actionable changed the paradigm of how clinicians see how we can treat patients, right? So we thought this is good. So now, I mean, for all the oncologists, they now have a molecular tumor board. Every single tumors will then be sequenced to look for the next driver oncologist. But does it really work? So recently, Daniel again published a work that's done within the Singapore General Hospitals. So among all the patients, so every single patient that come into Singapore right now, when we determine the confirmed case of tumors, we then send the tumors to molecular tumor board. We go through profiling, right, Foundation 1 or our in-house targeted panels to look for the next or the presence of actionable driver oncologies. Despite so, right, 785 patients it's close to 80% of all the individual cancer patients are found to have driver oncogenes. But yet, 62% are found to have not any treatments available. So it should highlight that this approach of targeted therapies has its limitation. So what can we do in the laboratory? So we started asking this question about 10 years ago. These are my students who joined the lab about 9 years ago. Can we look for another mutation that is specific to Asian? So at the point of time, we profiled a panel of our 50 squamous cell carcinoma of lung. We identified a CMAP variant at the CMA domain, right? We soon realized this is actually germline polymorphism. So it's found to be about 10 to 15% among Asian populations, higher in the Indians, lower in Malays. We have the luxury to work with pharmacology genomics in the bank in the NUH cohort. So what we collect is that we collect the blood from these individuals, cancer patients. We randomly sample 100 individuals from each cohort. I mean, contains lung, breast, lung cancers per se, right? And clearly that there isn't any enrichment of this germline polymorphism. So we know that it's not a cancer-causing genes like BRCA1 or BRCA2. But what is surprising to us is that we track this association of the variants to the survival of the individuals. We found that specifically only in head, neck, and lung squamous cell carcinoma, we are now seeing a more aggressive cancer phenotype because these tumors basically, all patients really have a poorer survival, right? So we thought at the point of time, hey, we found the next EGFR. So what do we do next? We immediately go and ask the question, can we test the drug, crizotinib, which is MET targeting tyrosine kinase inhibitors, right? Surprisingly, in the mouse model, if you compare the wild-type variant and the mutant variant, we are actually not seeing any effects of the crizotinib, and that's very surprising. But what we can be absolutely sure is that the tumors, when you're expressing the polymorphic variant, they are actually more aggressive. You can see that they're actually growing faster than the wild-type counterparts. So what is happening, right? So we go over to the drawing board, we perform more biochemical assays, mass spec, YP, many box, 1-2 years of work. We eventually realized that these mutations, despite having just one single codon changes in the CMA domain, it actually alters the structures of these variants. It is instead forming a homodimers as what CMAP usually functions. It now binds to HER2 as a heterodimers. It has a rapid firing of oncogenic signaling, right? So in the mouse models, we show that using different HER2 blockers, afatinib, herceptin, it all works beautifully well. We published a paper, we thought that's the end of it, right? But since this variant is so prevalent among Asians, and we have approved HER2 inhibitors in the clinic, so we have a luxury to actually work with two very prominent phase 1 clinical coordinators, Prof. Gobuncha and Prof. Daniel Tan. So we started working with Singapore Translational Cancer Consortium, which is the large consortium trying to bridge the gap between different hospitals to bring in more cases together. We enroll head, neck, and lung squamous cell patients. We screen them using digital job of PCR on the polymorphic variants. This will give us results within 3 to 4 hours. So we get a blood in the mornings, we get the results by noon, right? We immediately tell the hospitals what can we give to the patients, right? So this trial was actually completed around in January. So we don't have the data yet, I'm going to show it to you. But this is one of the cases that came in last year. This individual is a lady, relapsed on chemotherapy, relapsed on pembrolizumab, relapsed to any type of therapies, right? Very aggressive head and neck tumors. I hope I can convince you that within 2 weeks of afatinib and 6 weeks of afatinib, we are really seeing almost complete remission of the tumors. So it proves that this is really not the indicated treatment. Afatinib or HER2 blockers are not being used to treat this variant, right? But we're now seeing a significant response towards it. So now we're tracking the patient survival over time. But having this success, we then ask the questions, right? Can we now exploit the Asian-specific genome to better understand these issues? We have a lot of efforts in Singapore, particularly the precise cohort, the SG10K cohort. So this really is something that we are trying to profile. What are those Asian-specific variants that are not studied in the Caucasian populations? Right? What are those that are missing in UK Biobank? So I would like to give you one example, right? So I profile all those tyrosine kinases. EGFR is very popular, so we use EGFR as examples, right? So if we look into all those germline variants across the Asian populations, we are now seeing there's a lot of recurring between 3 to 5% of all these little somatic germline polymorphisms. And you'll be surprised that most of them actually present in the extracellular domain. So they're not found in tyrosine kinase domain as the usual LT8 mutations that is oncogenic. So for those who don't really understand or are not familiar with EGFR, the activation of EGFR basically depends on 2 very distinct mechanisms. First is that the tyrosine kinase domain must flip, right? So it must turn from this a V shape to an N shape, right? And next is that the extracellular domain must bind to each other. So these are the 2 key events. So the question now is that can we study the EGFR mutations or the structures to determine its activity? We look into our CMAP cases. It took us 10 years, almost 10 years, from identifying the mutations to getting the drug into the patients. It's too long. We did maybe 10 years, right? So can we now using machine learning? I'm trying to avoid the word AI. So can we use machine learning to help us to understand this, right? So how about molecular dynamics? It's really one particular technique that will allow us to use known protein structures and understand the force field changes to simulate the understanding of how these mutations affect the protein structures. So we are just basically using to understand the perturbations. If you have one conjugate variant, if you have one mutations, how does it actually alter the function of the protein? We work with our colleague, Chandra Verma, who is the research director for the Bioinformatics Institute in Singapore. We then understand, just look into the mutations that we have identified from the precise cohort, what do we actually get, right? So what we know is that this particular codon, the lysine 253, it is found in the ECD domain, right? And once it's related to the arginine, it actually introduced 3 additional hydrogen bonds. So it's predicted that this will actually improve the EGFR dimerization. Another conflict variant at the leucine 640, it is found at the N-terminus of the transaminase domain. And these particular variants are predicted to actually stabilize the leucine to isoleucine changes and allow the cells to be locked at the active state. Although there's this prediction, right, just pure predictions, but it gives us a possibility that there are actually more pathogenic variants across our Asian-specific variants. So this is impactful. So then we ask, we are researchers, so we ask, how can we improve the understanding of profiling this variant? So my students, Hui Ching as well as my then RA Grace, then asked a question, right? Can we now design a way to quickly introduce this mutation into the cells? So we have now in patent a 2-in-1 dumbbell system to rapidly allow us to modify a knock-in or introduce a different gene into the cells. And with this, we can then functionally profile these pathogenic variants, drug screening, and hopefully we can draw prognostication to our clinical cohort because we have all the clinical data that's stored as DNA bank. So I know my time is running out, so I'll try to rush through the second part, right? So the second challenge that we really identify is that we have all the trials ongoing, right? We don't have a large cohort like China, the Europeans, and Americans. So how can we identify a right predictor of response to any new drugs that we are bringing into Singapore? So let's use nasopharyngeal as an example. It's a hot tumor. It is being induced by viral antigen, EBV induced, right? We know that it's actually immune rich. So if you do, you take a samples of nasopharyngeal carcinomas, you do a single cell profiling, 70 to 80% will be all immune cells and stromal cells. So it's definitely immune rich. But why is it important? Because it's a very aggressive treatment. The only standard of care is still chemo-RT, but relapse rate is high, and overall survival is also very poor. So what do? We then ask the question, is immune hot tumor, right? So what should we do? We're definitely going to work in immunotherapy. So in fact, it's true, almost 40% of the trials right now on nasopharyngeal carcinomas are all immunotherapies. But yet, only 20% of this individual actually responds. So that is a major gap, right? It's immune hot tumor doesn't respond to immunotherapy, so something is wrong. So we ask the question, right? We know that VEGF, vascular endothelial growth factors, are highly expressed in the NPC microenvironment. So this VEGF is known to actually cause, right, the destruction of the maturation of the blood vessels. It causes leakiness. So it doesn't allow us to deliver the immune cells or the drug to the microenvironment, right? So we ask the question, if we give a VEGF-8 inhibitor, bevacizumab, Avastin, in other terms, right, does it actually help? Avastin chip now is off the patent, so everybody can use that, right? So the trial they published in about 3 years ago, I'll be a few minutes late, I'm sorry. Right? So we really shows that just giving one dose of Avastin, we are able to significantly increase the infiltration of CD4 and CD8 T cells, right? And this will actually lead to a significant increase in the survival. So by just combining Avastin and chemo-RT, we are increasing the complete response to almost 40%. And that itself is remarkable, right? This leads to the second trial that we just published about earlier this year. So what we do is that we have arm A, right? Drug A is pembrolizumab, the word is published, so there's no longer a need for me to hide the data. So drug A is pembrolizumab, PD-L1 inhibitors. In arm B, we basically prime the individuals with bevacizumab 1 day before we give pembrolizumab. So what is the response? And clearly only 12.5% actually react, and this is basically the same as what we get from the literatures, right? Less than 30% responded. But once we are just priming with Avastin, we are now increasing the response to 60%. And some of these patients, particularly this lady, she's still surviving after 6 years, and she always come to the clinic and give us chocolate, right? And so this is a remarkable response, right? So it published the work. But as a researcher, we never stop here. We always ask, right, what drives the immune trafficking into the tumors? Secondly, does blocking VEGF-A increase the activity of a T cell? So is it a quantity or is it a quality per se, right? Do we increase the quantity of T cells or are we increasing the activity of the T cells? We don't know the answer. So again, our Grace, who is now a student, or from our students right now, she then said, can we look into the plasma? Because we can't ask patients to come back for rebiopsy. It's a painful process. It doesn't give any benefit. So with that, can we take the plasma that we have, a serial collection of plasma that we have? Then she postulated that since Avastin is causing systemic changes, we should have a systemic biomarker that can predict a response, right? So how do we do this? We picked the technology called O-Link. So O-Link is a high sensitive, antibody-based, NGS-based proteomics assay that allows to profile the serum proteomes rapidly. So we get the result within 2 weeks. And so within this proteome, what can we learn from these patients? So the next slide is big. So yeah. So just to really recap with you. So arm A is only single arm, pembrolizumab. Arm B is pembro, right? So these are the good responders, bad responders for arm A. If you look across all the changes, you don't actually see much, right? But what is actually striking to us is that if you look into the good responders among the arm B, right after a single dose of Avastin, we are seeing global changes in the systemic biomarkers, right? And so Grace then asked the questions, are these biomarkers really important, right? So we compared those changes after 1 dose of Avastin. We look at the differential protein expressions. We are not able to get 800 upregulated proteins. And I can't review all these, but I can tell you there's actually key markers, key interleukins that actually prime the T cells and B cell activities. So this becomes a question is, are these a true biomarkers for us to predict potential response for these patients when given a combinational treatment? So just to quickly summarize, this will be the second last slide. Sorry. Right. So what are we doing in the lab? Right. We know that cancer is a very aggressive illness. It often causes relapse, metastasis. In the peritoneal, metastasis is nasty. It just goes into every single organ, right? If the lab is basically asking the question in a different way, liquid biopsy itself is minimally invasive. If we are able to use this plasma to understand what is happening in the tumor, it's a big plus to all of us. So one of the things we showed you earlier is that we extract the blood cells, we get the germline DNA, we look for what are the better ways to have targeted interventions to Asian populations. We ask for what are the better assays that we can rapidly translate into clinical trials to support the oncologists because they have no time, right? Do we have a quick assay that can inform the decision of how a target therapy should be designed? How we can complement the molecular tumor board instead of using a foundation one, it takes about 2 to 3 weeks to get the results back. And also from the serum or the plasma, can we now look at biomarkers for us to predict the response therapies? If the patient is not going to respond at this dose or this combination, can we save the effort to change to different agents for us to be able to intervene the disease before it progresses? So these are the work that is really focusing in the lab right now. And I acknowledge that my previous school, Young School of Medicine, I'm now in the Kong School of Medicine, trying to bridge the gap between 2 different medical schools. I would like to thank my collaborators, of course, the industrial partners, Darwin and Novogene. And so this is the small team that we have that we're working day and night to try to tackle these issues. And thank you, and sorry for being late.